what I'm going to do in this first talk this morning is to try to give a bit of a background into the kind of experimental data that we have, primarily from laboratory evolution experiments about evolutionary dynamics in microbial populations. And essentially what we're looking at in these kinds of experiments is, and the theory that's associated with it, the kind of theory that Gautam will talk a little bit about some aspects of in an hour, is understanding how evolution and evolutionary dynamics works as a sort of stochastic algorithm. There's some sort of, in any kind of evolutionary situation, some sort of set of possibilities, essentially a fitness landscape on which the evolution is taking place in that particular organism, in that particular environment, at that particular time. And somehow these kind of dynamics of the evolutionary process lead us to some patterns of outcomes that we observe in the experiments or, of course, in the natural world. And, we get data sort of all different types. I'll talk about a variety of those types as we go along. But you see things like this diagram at the right, which is a figure from a paper by Olivier Tenelion, about 10 years ago now, which I happen to particularly like, where he evolved a little over 100 independent E. coli populations for 2,000 generations in response to high temperature stress, and then sequenced each of the evolved populations. And what you're seeing here is each of these rows is some evolved population, and the different mutations that were found in at least one clone isolated from that evolved population after the 2,000 generations of evolution. And the different colors represent different types of mutations I don't want to get into in great detail, but you see these kind of patterns of evolutionary outcomes in these lab experiments, which obviously reflect something about the combination of deterministic and stochastic factors that characterize evolution on this particular landscape in this particular population. So you see some mutations that are repeatedly observed across many replicate populations, a lot of other complicated patterns of partial repeatability. And one of the things that's sort of remarkable to me, having worked in this general area for 15, 20 years now, is that how this stochastic algorithm works and how to characterize, how to look at these outcomes that we observe and infer something about the underlying landscapes remains pretty poorly understood. So, all right, what I'm going to do now is just start by talking about a few different, I'm going to talk about a few different kinds of data that come out of these experiments and some of the general conclusions that are observed repeatedly across multiple systems. So the basic laboratory evolution experiment that people do is you take some microbial population. People also do work in C. elegans or Drosophila or whatever, but I'll talk about microbes. And you put it in some flask of media, and you let them grow, and then you dilute them after some period of time into a new sterile flask full of media, and you just repeat this again and again and again and again. And there's lots of experiments along these lines. Here, I'm going to talk about arguably the most classic such experiment, the long-term evolution experiment conducted by Rich Lenski and his collaborators at Michigan State University, where this has become particularly well known because Rich and colleagues have been doing it for a very long time, over 30 years now. Basically, they have 12 replicate populations of E. coli. They're evolving it in this standard batch culture way. Just to note, this leads to a sort of fluctuating population size from day to day. Basically, they do, in this experiment, they do a 1 to 100 dilution every 24 hours. And so the population grows about 6 2/3 generations per day on average, and then gets diluted down by a factor of 100 and grows again, so on and so forth, which in this experiment leads to an effective population size of roughly 30 million. As I said, this has been going on a long time, over 30 years now. It's just actually past 70,000 generations pre-pandemic. It's shut down at the moment for obvious reasons. I'll talk about data from others as well as some data that my lab produced roughly in the 50 to 60,000 generation, through the first 50 to 60,000 generations. And they've frozen these populations every 500 generations. So one of the really nice things about these experiments in microbes is that you can just, store populations in glycerol stocks indefinitely. And so then you have a sort of frozen fossil record that you can go back to and recapitulate things that have happened over the evolutionary history. So we have this, 60,000 generations, now 70,000 generations of evolution with time points frozen every 500 generations. And one thing that you can do is you can look at phenotypic changes. And so people look at all different kinds of phenotypes. Of course, the sort of most relevant phenotype in these experiments is the competitive fitness of the populations. And that's a sort of complicated quantity and not really necessarily a simple one-dimensional quantity. But one way that you can measure it is essentially you unfreeze a sample from one time point and you unfreeze a sample from the ancestor, from this generation 0 time point or some other time point, whatever you like. You label one of them with some marker, some neutral marker, and then you compete the two in the conditions of the evolution experiment, and you see which one outcompetes the other and how quickly that occurs, and that's a measure of the relative fitness of the population. And you can do that at different time points to see how that relative fitness is changing over time. And what you see in this experiment, these colored lines are the replicate populations, the 12 replicate populations, this black is the average, is something that you very typically see across lots of these experiments. You get a fairly repeatable, predictable pattern of fitness change over time in these replicate lines. You get rapid increase in fitness, roughly 20% or so over the first few thousand generations, and then it sort of slows down. It may not be obvious from looking at this, but the fitness does continue to increase, and later data since this paper was published a half a dozen years ago show that continues through at least 60,000 generations. You get this sort of slow, steady increase over time. So we have this kind of predictable phenotypic evolution characterized by sort of rapid adaptation in the beginning and then sort of declining adaptability, although continuing adaptation over longer timescales. And this is this is one example from this experiment, which is the most well-known, but this theme has repeated again and again and again across many evolution experiments in many systems by many labs. So this is a very typical thing that you see.
Michael, sorry if you already said it. I didn't quite catch how that's quantified, the fitness. What does this percentage mean exactly?
This percentage basically means that what you do is you take the unfrozen population from whatever time point that we're looking at here, say an unfrozen time point from generation 21,000 or something from each of these populations, and you take an ancestor that has been labeled with a neutral marker, you mix them together in the exact conditions of the experiment, and you grow for a few days, okay, according to this standard protocol. And every day, using the neutral marker, you measure the relative frequency of the ancestor with the evolved population.
Perfect, thanks.
Right? And what you'll typically find is that the evolved population will increase exponentially, okay? And so if you plot the log of the frequency as a function of time, you will typically get a straight line. And the slope of that line is this relative fitness.
Great. So it's the ratio of the exponential rates of the...
Exactly. And I don't want to get into, there's a whole other talk we could have about the problems with that. And right, it doesn't have to be exponential increase. There can be frequency dependent effects. There can be non-transitive fitness interactions. All these things do happen and they're important and interesting. There's whole other things that we could be talking about here. But, to a sort of first order sense, this is what happens. Things, it's not surprising. You evolve something in some environment, things get better. Okay. So that's what happens in terms of the phenotypic evolution. You have this predictable pattern, which is characteristic across this experiment and many others. So a thing that folks have started doing over the past, maybe 8-10 years or so is looking at what are the genetic bases of this phenotypic adaptation and how predictable is the genetic basis of the adaptation. And this, of course, corresponds to the now ability, our ability now to sequence populations and observe how specific mutations have accumulated through time. And so I'm going to talk about work that my lab did in collaboration with Rich Lenski's lab and Jeff Barrick's lab, a couple of other labs, to look at this within the LTEE, but this same kind of theme again repeats again and again across many other evolution experiments. So basically what we did was take every single frozen time point through, at the time we did this work, there were 60,000 generations. So this was 120 frozen time points at 500 generation intervals. And we just did sort of whole population sort of metagenomic sequencing. Okay, of each of those populations. And so the kind of data that you get is, I'm just sort of zooming in on the first 10,000 generations of one of the populations. I'll show all of the rest of them in a moment. Here's an example of the kind of thing you see. All right, initially there's nothing, you start from a clonal population, and then after 500 generations, you sequence the population, and what you observe is that there are some mutations that have spontaneously arisen in that population. And some of them have gone up, these couple of mutations have gone up to 50% frequency. So at this point in time, half of the individuals within this population have this mutation. And about a third have these mutations, and 20% have this mutation, and so on. And then you sequence the next time point, and what you see is, oh, this mutation, which I'm indicating by this blue line, has gone up to 100% of the population, and so has this one. These ones have gone extinct, and new mutations have arisen. And we can sort of, follow this through time. New mutations are constantly arising, and some of them are spreading through the whole population. Some of them are fluctuating around for a bit and eventually going extinct. And this process continues. And you have, underlying this kind of predictable phenotypic evolution, you have this kind of huge mess of stuff happening. Lots of different mutations arising, some spreading to fixation through the population, others going extinct. And if we look across all the replicate populations, here are 6 of them. Now I'm showing 60,000 generations here, and these 6 panels are 6 completely, 6 of the independent replicate populations. And so each of these lines is a sort of new mutation that arose in that population. I'll talk in a few minutes about what those mutations are, okay? But for now, the thing you can sort of see, at least at a first order, is that, there is a lot of stuff happening, okay? There appears to be quite a bit of differences between the replicate populations. They're not all doing the same thing at some level, in some phenotypic, in this, at the genetic level. And there doesn't appear to be this pattern of sort of slowing down of evolution over time that we saw in terms of the fitness trajectories. If you recall, the fitness increased rapidly at first and then sort of more slowly going on as we got later. If you look at this data, to me, it just sort of looks like there's a kind of a steady pace of genetic change, okay? Here are the other 6 populations, even more different than the first 6, and all kinds of wild stuff happening here. These 6 populations, at some point or another, mutations arose and spread that change the mutation rate, that increase it. And so, here is the clearest example at the top, where one of these mutations, oh man, I'm forgetting which gene now. I believe it's MUT. That arises in a mismatch repair gene, basically knocking out a mismatch repair gene and spreads. And then after that, you've got just this whole wall of color. It's just because there's so many mutations arising in this population. And okay, so I could talk for a long time about this. I won't, but I just want to sort of emphasize the sort of key points of this that are common across many other experiments. This is obviously the longest laboratory evolution experiment that we have available, but the same kind of patterns repeat again and again in lots of other systems. Again, this here is just replotting the change in fitness of these populations over time, where you see this fairly consistent pattern, okay, between replicate populations. They all sort of increase rapidly at first and then slowing down over time. At the bottom here is the sort of pace of mutation accumulations, the sort of rate at which mutations fix. And here you see this is also a kind of a steady accumulation of new mutations over time, okay, in each of these replicate populations. And occasionally what you see is these populations that shoot up off the scale. And here in the inset is sort of zooming out to look at those. Those are the populations in which the mutators accumulated. And so you see these kind of steady patterns of mutation accumulations and fairly predictable patterns of phenotypic evolution. But just to sort of emphasize then, you know, we see this sort of wild dynamics at the genetic level, and not at least at a sort of gross visual level, these populations, they look quite different from one another.
I have one naive question about the previous slide. Like, what does it mean to see like a group of mutations moving together, like up and down in the previous slide? Are they like phenotypically related or?
No, what it means is that these are clones, essentially, presumably, where you have some clone of individuals that has accumulated multiple mutations. And that clone of individuals will either spread and take over the population, and all those mutations will go to high frequency, or it will go extinct and they'll all go down. And so you see this sort of competition often between clones that drives groups of mutations up and down. And that's the signatures that you're seeing there.
I see, okay.
On the fitness plot, I believe on the next slide, I had a question. I wonder, maybe just from too few points, I'm maybe extrapolating a pattern, but I wonder if the noise here could be considered like heteroscedastic in terms of its like increasing over time. And I wonder if you think that's a potentially interesting pattern that's general.
Yes, so in this particular experiment, right, it was started in 1988. And so the kind of markers that are used to measure fitness are not really ideal. And so you do definitely have, there are all kinds of complicated issues associated with the fitness measurements. And certainly there is heteroscedastic noise where the noise in fitness measurements is correlated with the fitness because of the way that the counting is done, essentially. Like when things go down quickly, then there's more noise. And and there are efforts to sort of improve. I don't think that the overall conclusion that I'm sort of trying to draw here is affected by that, because I mean, then people have done more careful things. Like you don't have to compete against the ancestor. You can construct other kinds of, other references, and people have done that and get more accurate measurements that way. And the sort of basic message is the same. More recent experiments, use better marker systems that allow you to measure these things much more accurately. And you still have heteroscedasticity in the fitness measurements, like that in the noise. That's always going to be a sort of feature of this kind of measurement, but it's a lot better, and the sort of general conclusion is the same.
One more quick question.
Yeah.
So even among the populations that don't become hypermutators, there seems to be about a twofold variation in the steady slope. Is that interesting at all?
Yeah, I mean, it seems interesting to me. It's weird to me, right? It strikes me as, it's interesting, and I don't understand it.
Could there be weaker effect things that change the mutation rate slightly?
There absolutely could be, right? So, I mean, measuring mutation rate to, 20% is difficult. And so, yes, there could absolutely be weak effects on mutation rate. I believe this blue line here, this guy has some weak effect IS mutator. So it has basically an elevated rate of a certain kind of insertion sequence hopping around. And that is at least partially responsible for why this guy is above. In terms of these ones that differ by maybe 10 or 20%, we haven't found anything. There's nothing, I mean, we know what the mutations are and we don't see anything obvious, but I don't know why. But I mean, to me, it's sort of interesting that like even these guys here that clearly have dramatic mutator phenotypes that go up by a huge factor, you can't really pick them out of this top diagram, right? They have this dramatic change in the rate of mutation accumulation by like a factor of 100, right? And yet, they don't really adapt much faster or slower. And it's kind of weird. Like another thing that's really weird to me, I mean, we could go on for a long time about stuff that's weird and unexplained. Another thing though that's really weird to me is that half of these populations get these mutator genotypes that fix. And if you think about it, these are populations with an effective size of about 30 million, and there's 60,000 generations. There are like hundreds of thousands to millions of independent mutation events that create mutator alleles. Okay, so like these mismatch repair genes are knocked out like 100,000 to 1,000,000 times over the course of the evolution of each of these populations. Okay, so it is not like the case that the 6 populations that in which we get these mutator alleles fixing, that's because it's a rare event and it only happens occasionally and it just happened to occur in these 6 populations. It's happening all the time in all 12 populations. And you would kind of think this is like one of those things, it's like either good or it's bad, right? Presumably the mutators, they may have some direct physiological costs and benefits. And then of course they increase the rate at beneficial mutations arise in those populations, but they also increase the deleterious load. And you might imagine, okay, there's just some balance between those factors, and it's either good or it's bad. And if it's good, you'd kind of expect them all to do it. And if it's bad, you'd expect none of them to do it. And instead you get half.
Right, so it's balanced enough that it's kind of like,
yeah, it's kind of wild, right?
Yeah.
And it's balanced enough, you get, obviously these mutators, I mean, they're accumulating a lot more mutations, presumably more beneficial and more deleterious ones, but somehow it's in balance such that you really can't pick them out of this top plot.
And just for intuition, like size of genome and mutation rate, should we be thinking infinite genome or is it more like lots of recurrent and back mutations?
So it's in between, but I guess closer to the first. So the genome size the genome size changes a bit over the evolution, but it's like, 4 million base pairs or so. The per base pair mutation rate is on the order of maybe 10 to the minus 9.5. So, you're looking at, one mutation per few hundred generations per individual, but then you have a population size of 30 million. So, I mean, you can do the math here. We do see recurrent mutations and back mutations. They do happen, but they're, in terms of them spreading to high frequencies, it's relatively rare.
Thanks.
Okay, let me just say a few words about what these mutations are, which sort of emphasizes sort of more of these kind of ideas. So, okay, if we look across, I'm just looking here across the 6 non-mutator populations, and we know, of course, what all the mutations are, and you could classify them at various levels of organization. So here I'm looking at what genes those mutations are in. It's probably kind of hard to see. I don't want to belabor the details of it, but again, this sort of gets to to the stochasticity of evolution at the genotypic level, we see what I'm showing here. Okay, this at the left, mutations in the gene MAL-T, we see 18 examples of them across the 6 non-mutator populations. So an average of 3 independent mutations in that gene per population that reaches a sort of detectable frequency that sort of gets to close to fixation. And okay, there are others, PYKF, NADR. And so there are a few mutations, few genes in which we see mutations again and again, but then there's this sort of huge spray of stuff that we see occasionally, okay? And, this is just an example of when the mutations occur. I don't want to get into this in great detail because there's other things I want to talk about, but the general conclusion that you can sort of get from this at some levels, you might imagine a picture of evolution in these systems where you have some sort of, you start from some genotype over here, and there's some fitness landscape, and like this is some simple laboratory environment. You might imagine there's some optimal genotype, some fitness peak over here. And these populations are just climbing up this mountain. They're accumulating some set of unambiguously beneficial mutations, and they're approaching this optimal genotype, and they're kind of all doing the same thing. They, are trying to accumulate the same set of beneficial mutations across all these replicate populations. And then they're going to get to this fitness peak, and that's going to be the end. Okay. And I don't have time to really get into all of the evidence for this, and Gautam will presumably talk about some of the theory behind, so I don't want to preempt that. But suffice it to say that the evidence seems to be that doesn't really happen in this experiment or across any other laboratory evolution experiment that we've seen. All these replicate populations appear to do, somewhat different things. They go in different, they accumulate different mutations. There's evidence that, often if you get one mutation first, then there's a different set of mutations that's beneficial going forward from that. Whereas if you had got this other mutation first, then you go in this other direction. There's this kind of epistasis and contingency whereby what mutations happen first either open off or close off other pathways. There are some mutations that, tend to be sort of low-hanging fruit, sort of everybody gets them right away. They're like sort of unambiguously beneficial, and they're sort of like, okay, everyone wants to get that. But most other things are not like that. There are mutations that, they're beneficial, seem to be beneficial in some contexts, but not others. There's not a clear sort of fitness peak, where all the populations are trying to accumulate this set of 10 mutations, and they all get some, as they get more and more fit, they get a larger and larger combination of them. Okay, so I want to talk a bit, all right, I'm going to sort of skip over that. All right, so I want to talk a bit more about that, okay? And so there's a bunch of other kinds of experiments that people have done to try to get at these questions of what does that landscape look like that's leading to these kinds of patterns that I was just talking to you about. One kind of thing that people do is, you take some evolved population and you sequence it and you see, okay, I got this set of mutations. And then I make all the combinations of those mutations and I look at what the landscape is. So it's kind of like the landscape of the mutations that actually accumulated along the line of descent in one evolved population, okay? Which is, if you think about it, a kind of a weird way to look at a landscape, but nevertheless, it's something that folks do. Here's an example. There's a whole bunch of papers. This was kind of all the thing 10 years ago. They all kind of come to similar sorts of conclusions, although there's all kinds of details that differ. Here's a figure from one of them. I think it's from this paper by Chris Marks's group, where they took 5 mutations that accumulated in one of these Lenski lines over the first 1,000 or so couple thousand generations. These mutations, they called them R, T, S, G, and P respectively. And so there are 5 of them. There's the ancestor, there's the fully evolved genotype, and then they made all 2 to the 5th combinations of these 5 mutations and plotted the relative fitness here. And we could go on about what you see in these plots, but the bottom line is if you sort of look at this landscape, there's all kinds of epistasis here. There's mutations that are beneficial on some backgrounds and deleterious on the other, right? Overall, you want to get all 5 mutations, but the order matters. If you get them in the wrong order, then you kind of can get stuck in various ways. And so this sort of result came up in a number of different independent experiments of this type, where you get lots of epistasis along the line of descent. And at some level, it's sort of inconsistent with the observations of predictable, or at least it seems inconsistent with the observations of phenotypic predictability that I was talking about a few minutes ago. If there's all this kind of epistasis where you can get trapped on local fitness optima, then why, when you do many replicate populations, they all kind of do the same thing from a phenotypic level? So a concept that I've been trying to promote is this sort of distinction between what I like to call microscopic versus macroscopic epistasis. And really what this comes down to is that when we think about fitness landscapes, our natural sort of intuition is to think about like a landscape that we're familiar with from our everyday life, you know, hiking in mountains in New Hampshire or something, which is a fundamentally like a 2-dimensional or 3-dimensional, if you like, landscape. But of course, genotype space is much, much, much higher dimensional than that. And so a lot of our intuition can be off. And so, you know, the way I like to think about these kind of landscapes is at any given moment, you only care about local things, right? A population is sort of some cloud of genotypes, which is sort of held together by common ancestry and is sort of centered around some sort of location in genotype space. And it explores locally around that. And what kind of matters is what are the local prospects? And, you know, as you adapt, you move that cloud through the genotype space. And what matters is not sort of the epistasis or the interactions sort of along the line of descent, but what matters is how the properties of the local environment changes as the population moves through genotype space. And so, you know, a way to think about that is the most local aspect of that. This is not the only thing that matters, but like just to crystallize this a little bit, the most local aspect of the landscape is if we say we make point mutations, single point mutations, what is the distribution of their fitness effects, right? And so at any given moment, you know, you have some genotype of some clone in the population, and there's some distribution of fitness effects of all the possible point mutations that it could get. And then we ask, okay, let's say it gets one of those mutations. How does that distribution of fitness effects change? Okay. And there are a couple of possibilities, right? Like a natural sort of thing is that, you know, depending on how I phrase this, people have opposite intuition about what's obvious. Okay, which is, I think, kind of nice. But we know that there's like lots of epistasis along the line of descent. So often you might get a situation where like, okay, this is the distribution of fitness effects of beneficial mutations from that genotype. And the gray mutation occurs, you might have a lot of pairwise epistasis where you got this gray mutation and now this blue mutation is no longer beneficial, the yellow mutation is no longer beneficial, the white mutation, now instead of being strongly beneficial, it's weakly beneficial, and the black mutation, instead of being weakly beneficial, it's now strongly beneficial. But the overall distribution of fitness effects doesn't change. Okay. We know there's a lot of pairwise epistasis, and so we know that things are changing around in this distribution as you get individual mutations. But they could be changing around in such a way that the overall distribution doesn't change. Or they could be changing around in such a way that it does. Okay. And you could argue about which of these you think is more natural or which is not more natural. I don't know. I mean, I get different things. I mean, on the one hand, people like to say, well, how could it be the case that lots of things are changing their effects, but the overall distribution doesn't change? So they view this top scenario as improbable. But then on the other hand, people sort of say, well, how could it be the case that you get one mutation and it totally changes the distribution of everything else that's possible? Okay. But anyway, this is an empirical question. And so we can sort of characterize epistasis in this way, is like sort of how does the local properties of the landscape change as you evolve? Okay. And we can, here I'm showing like the distribution of fitness effects of beneficial mutations, which people often kind of refer to as how adaptable is the population. One could imagine thinking about the distribution of fitness effects of deleterious mutations. How does that change? And Gautam will talk about this as well in a little bit. Okay, so these are empirical questions, and these are things we can actually measure.
Could you imagine a multidimensional phenotypic space as well with some components of fitness?
Sorry, I didn't catch. Can you repeat that?
Is it useful also to imagine a multidimensional generalization if there are several components of fitness? Or
yes, absolutely. So I'm not going to talk about that at all, but absolutely. Like, so for example, right, in these Lenski lines that I was talking about earlier, right? There's a complicated growth dynamics, right? There's some lag phase, exponential growth, there's saturation phase. There are cases where you get frequency dependent interactions and coexistence because, there's mutations that affect one of these phases, but not the other, and then they can interact in different ways. There's also sort of even more complicated things where like some strains, for example, you can get mutations where it increases the rate at which you consume glucose, but the side effect is that you leak more acetate. And then there are other mutations that you get which kind of specialize on the acetate, and you can get coexistence of strains that eat glucose fast and that scavenge up the leaked acetate. So yes, absolutely. Like we could get into all this kind of stuff. And really, like, right, fitness is this sort of more complicated, multidimensional thing. And of course, in addition to that, the whole environment can fluctuate, right? Like as you adapt to one thing, you're accumulating mutations that have pleiotropic consequences for other phenotypes. And, if you change the environment, you might change the relative balance of what's important and what's not. And those are all fascinating questions. And, I have 45 minutes. So, but you can get at them with the same kind of approaches, right? I'm just not.
Yeah, I was just the idea of going to the distribution, macroscopic picture,
right?
In general, anyway.
So yeah, you can absolutely like do things where like, okay, we're going to define various components of fitness or fitness in different environments, and we're going to look at as you get mutations, how does that joint distribution of effects, right? So I often like to think about that. It's just like there's actually, a joint distribution of effects on all different kinds of phenotypes. And as you evolve, right, and so I'm oversimplifying by talking about like one dimension of that. But, hopefully you can sort of, the same kind of principles, we would be talking about the same kind of things in that more general context.
Yeah, makes sense. Thanks.
Okay. So, okay, I want to wrap up in the near future. So I just want to talk about then briefly a couple of experiments to directly get at this question of macroscopic versus microscopic epistasis and how these local landscapes change. And then hopefully Gautam will talk about that in more detail and talk about the theoretical frameworks that he's developed to explain the kind of themes that we see in these experiments. So basically the kind of experiment that we can do to look for this kind of thing, we want to ask whether nearby genotypes have landscapes, local landscapes that are similar or different, right? Like if you get a mutation, does the local landscape change? So the way to do that is you make a bunch of related genotypes that differ by a few mutations or maybe by a lot of mutations, and then you measure the landscape around them. Okay, and so how do you do that? Well, there's a variety of ways that people have done this. So I'm going to just briefly talk about a couple. So here's one. We started from a single clone of yeast. Okay, my lab, we do mostly yeast experiments, so I'm talking about that. But other people have done similar things in E. coli and other organisms, and the basic themes, again, are the same. So, okay, we start with some clone and we evolve it, okay? In this particular experiment, we took 64 independent replicates. I'm just showing 7 here, and we evolved them, 64 independent things, and they accumulate a few mutations, just, and they're different because, they're independent replicates, and so they go in different directions initially, just by chance. And we get a bunch of these different found genotypes. And then we took each of these, okay, we made a clone, and we evolve a bunch of replicate populations, 10, I'm just showing 3, but 10 replicate populations from each of these. And so then we can ask, basically, do the replicate populations descended from this guy differ systematically from the replicate populations founded from this guy? Okay. And if so, that's sort of showing, obviously there's some variation between the replicates from one of these genotypes, just because there's chance in evolution. But we want to ask, above and beyond the natural variation between replicates founded from the same founding genotype, are there systematic differences between the replicates founded from this founding genotype and the replicates founded from this or this or this founding genotype? So in other words, to what extent does the founding genotype affect the future evolution? In other words, how do these first few mutations constrain what happened, the landscape of what's possible going forward? And I don't want to sort of get into this in great detail because I don't have that much time, and Gautam will talk about this presumably going forward. But basically the upshot is that the answer is that yes, these first few mutations do constrain what happens going forward. And they do so via this sort of predictable pattern of what we call diminishing returns epistasis, which leads to a sort of convergent fitness evolution. If those first few mutations you get leading to the founding genotype happen to be a little worse than average, so the founding genotype is sort of less fit than average, then those populations will adapt faster going forward. And if those initial mutations happen to be abnormally good, those populations will adapt more slowly going forward. So what that kind of means is that, if you sort of get behind at first, you tend to catch up. And if you tend to get ahead at first, you tend to sort of lag behind. And so there's sort of everything sort of happening to sort of lead to convergent fitness evolution. And, you could imagine that this is just because there's 5 mutations that you want to get that make you better. And, the guys that lag behind, they just didn't get any of those mutations. So then, of course, they can adapt quickly because they can acquire them in the future. And the individuals that adapted quickly at the beginning, they got several of those mutations. So then they can't really adapt anymore because they got those already. And the funky thing is that that's obviously sort of inconsistent with the picture from the Lenski lines that I showed you earlier. And it's also when we look at this more closely, it does not appear to be what's happening. What's happening instead is that, it seems like there's a really large collection of mutations that are just beneficial across all of these environments. So here are 3 examples of 3 mutations, basically deletions in genes that appear to be beneficial across all these different background strains. And this HO is a kind of a neutral control. And what we see is that, there's a collection of mutations that are beneficial across many, many genetic backgrounds. And, but they become systematically less beneficial as the genetic background increases in fitness. And so you have lots of beneficial mutations, but there's a sort of general pattern of diminishing returns where they become systematically less beneficial as the population increases in fitness. And so we and others have, looked at this in a variety of different systems. So another thing you can look at is, let's imagine constructing different sorts of sets of founding genotypes, okay? So we constructed sets of founding genotypes constructed by, that differ by tens of thousands of mutations by taking 2 divergent strains of yeast and crossing them and getting, a set of diverse sort of segregants out of that, right? And so these, and doing the same thing, okay, so we get, we cross two different strains of yeast, we get a pool of recombinant offspring that differ on average by tens of thousands of mutations, and we get the same kind of pattern where the populations founded from the lower fitness segregants, they adapt faster, systematically across multiple different conditions via the same sort of pattern of diminishing returns. And the last thing that I'll talk about briefly, okay, I'm going to sort of skip over this because I'm running low on time, but I just want to show this last slide. This same sort of pattern holds if we look at deleterious mutations. So there's sort of another experiment that we've done where we've constructed a set of specific transposon insertion mutations in a set of about what was it, Gautam? 175 or so different genetic backgrounds. And we look at the fitness effect of each of these mutations in each of these genetic backgrounds. And so what I'm showing in each of these plots, these are just an example of a dozen of these different insertion mutations that we constructed in each of these 175 backgrounds. As we might expect, this mutation has essentially no effect on the fitness in any of these genetic backgrounds. And here we see an example of an insertion mutation in a particular gene that has a deleterious effect, and the effect is fairly consistent across all genetic backgrounds. But the much more, and we see some examples of this, but a more common thing that we see is that these mutations, they have, these insertion mutations, they have effects that vary across the different genetic backgrounds. And they tend to vary in this kind of systematic way where they become systematically less good in more fit genetic backgrounds. And in some cases, even they're beneficial in the least fit backgrounds, neutral in intermediate fitness backgrounds, and then they become deleterious in more fit backgrounds. So there's sort of consistent patterns of this kind of effect. It's not universal, okay? There's obviously exceptions. There's cases where mutations have less deleterious or more beneficial effect in more fit genetic backgrounds, but the sort of overwhelming pattern is this kind of diminishing returns, okay? And so if you think about it, this kind of pattern can lead to this sort of consistent pattern of fitness evolution, sort of rapid phenotypic adaptation at the beginning of experiments and then slowing down over time, where different populations are doing very different things at a genotypic level. Okay, so that's the background I wanted to talk about and the kind of themes, and hopefully this sets up well for the kinds of theory that Gautam will talk about shortly. I just want to acknowledge folks from my lab who did some of the work from my lab that I talked about, as well as hopefully I've mentioned all of the collaborators and other labs whose work I've mentioned at various points.
Thank you for the invitation. I'm an NSF-Simons fellow at the Quantitative Biology Initiative at Harvard. My background is in physics and biology. My prior PhD work was in animal behavior and navigation, so something quite different. And I was working on problems related to, for example, how birds soar. That is, how do they use atmospheric currents to gain height. I was also interested in how animals track ordered trails. And more recently, I was working on methods to infer behavioral algorithms from behavioral data. Anyway, what I'll be talking about is, of course, something quite different from all of that. The appeal to me is that when we think about the evolution of an organism, we can imagine that the organism is essentially a complicated machine, right, with many moving parts. So one would expect intuitively that if you want to predict how exactly this organism will evolve, is going to be very difficult or pretty much impossible to predict at the microscopic level. On the other hand, as Michael showed, there seem to be quite a bit of predictable patterns when you look at the phenotype, right? So the broad question here is, how much of phenotypic evolution can we actually predict, and why can we predict anything at all? And most of what I'll be talking about is in this preprint over here, which is a theory paper, but it's based on many of the experiments, including the ones that Michael talked about, especially the ones done by Sergey and Milo in Michael's lab. Feel free to ask questions. I can't really see the chat here, so just interrupt me and ask questions whenever you can. Now, so I realized as I was looking through Michael's talk that I'm going to be describing actually the same experiments, but it's helpful that Michael went through the actual details of how these experiments are actually done, right? So when we think about predictability in phenotypic evolution, the classic experiment that comes to mind is Lenski's long-term evolution experiment in E. coli. I'm not going to go into the details here. Michael already did that. And the idea is essentially that Lenski drew 12 replicate populations of E. coli over the last 32 years. Right now, it's at around 70,000 generations. And what you're seeing here is the fitness relative to the ancestral strain for the first 50,000 generations, right? Now, Michael showed some of this data. These evolved populations have since been sequenced, right? And as you saw, the dynamics of molecular evolution and the specific mutations that arose and got fixed in these across these different replicate populations is quite diverse, right? On the other hand, what you see are these repeatable patterns in fitness evolution, right? So for many of these populations, as we saw, there are like half of them were mutators, half of them were non-mutators. But on the other hand, for all of them, we see that there is some rapid adaptation in the beginning, followed by slow but steady rate of adaptation as you go forward. Right? Now, these observations have been reproduced in many other experiments, usually at a much smaller temporal scale, but perhaps more at a larger scale in terms of the number of variants actually evolved. So for instance, this is the one, this is an experiment in yeast from Michael's lab, which he described as well. Right, so in this experiment, 640 different populations of yeast were evolved for 500 generations, right? And of these 640 populations, there were essentially 64 different variants and 10 replicates of each variant. Right? I'd like you to pay attention to basically two facts here, right? So this is, what you're seeing here is the fitness gained over 500 generations versus the initial fitness of the 64 different variants. And you can see that there's a wide variation in the initial fitness, about 20%. And
can I quickly interrupt you?
Yeah.
Hi, I'm Phil, Moffitt Cancer Center. Can you go back and define fitness for us?
Yeah. So fitness here, first of all, it's fitness relative to the ancestral strain. And there are different ways to measure fitness, but in this case, what is being done is that it's a competitive assay, and if you look at the number of these variants over time, they're essentially exponential curves, and the fitness here is defined as the difference in the growth rate of these two different strains that you're competing.
Using how many time points for the difference? 2 or more?
So in this case, I believe they were when you're measuring fitness, they're grown for around 5 days, around 40 generations. And Michael, correct me if I'm wrong, but I guess there are 5 time points.
So it's relative exponential growth rate.
Yeah, this particular experiment, I think we do different things. I think this experiment may have been 4 or 5 time points. And basically what you do is you plot the relative frequency, the log of the relative frequency of the evolved strain versus the ancestor. And typically that's a straight line, and then the slope of what you're measuring is the slope of the line, and that's the relative fitness. So it's sort of the relative exponential growth rate of the evolved versus the ancestor.
All right, thanks.
Thank you, Michael. Right, so what you're seeing here is the fitness gained related to the ancestor versus the initial fitness of the strain of the different variants. And there are two observations here that I'd like to highlight. The first is that most of the fitness gained seem to be explained by the initial fitness of the organism, right? So, I mean, for instance, you can have strains that differ quite a bit in genotype, but have the same fitness. But it turns out that the final fitness after these 500 generations of evolution seem to be pretty much explained by the initial fitness. I mean, you can do this R squared. It's about, I guess, around 80% of variance is explained by the initial fitness. The second striking observation over here is this negative trend that you see here, right? So the strains that initially had a relatively low fitness adapted much faster, right? So there's this general trend of declining adaptability, and the idea that fitter strains adapt slower. Now, if you think about the process of adaptation in a very simple version of what's going on, we can think of this simply as, I mean, you have essentially a mutation that arises in your population. This mutation has some fitness effect, right? We can imagine that this fitness effect is drawn from some distribution of fitness effects. Presumably, this distribution of fitness effects depends on the evolutionary history. And depending on the fitness effect, this mutation is either selected or it goes extinct. And if it gets fixed, the fitness changes. Of course, this is a very simplified version of what really happens, as Michael just explained. I mean, you have competing clades, you have clonal interference, you have hitchhiking, and so on. But for the purposes of this talk, I'll be focusing specifically on the distribution of fitness effects and how that depends on evolutionary history. Now, the advantage of what we can do in essentially the last decade is that we can evolve these populations in the lab. We can sequence them, and we can identify the specific beneficial mutations that arose during the course of evolution, right? So it means that we can identify the mutations and then measure their fitness effect on different backgrounds. And the hope is that we can kind of quantify how this distribution of fitness effects looks like.
So history in that slide means genetic background?
I'll get to that later, but history essentially, I mean, history in this case would be the specific sequence of mutations that have been fixed in a particular lineage over the course of evolution.
Right, okay.
Yeah. Which is, of course, I mean, which means that they have a different genetic background, different lineages have different.
Yeah, I guess I meant the genetic background right now of the, I mean, I guess I think that makes sense to me. Thanks.
Right, so this is precisely what they did in the experiment in yeast I explained a few minutes ago, which is that they identified 3 beneficial mutations. It turned out that these 3 were loss of function mutations, right? The pink one here is a control. And what you can see here is that the very same mutation, when applied on different backgrounds, has a different effect. So this is some kind of epistasis, of course. The striking observation here is that the very same mutation seems to have a much larger beneficial effect on less fit backgrounds. And since it's a negative slope, I mean, this is an example of diminishing returns, right? So you have this very same mutation. If you apply it on fitter backgrounds, it seems to have a lesser effect. And you also see, at least this data is a bit noisy, but you do see some kind of linear trend over here. More recently, this is the last experiment that Michael was describing. this linear trend of the fitness effect as a function of the background fitness is reproduced for many other mutations, including mutations that are on average deleterious. So in this experiment, what Milo did in Michael's lab was to generate about 150 different F1 haploid backgrounds, which means that these backgrounds were generated by mating two strains of yeast. In this case, there were the standard baker's yeast and a wine strain of yeast. These two strains differed by around 40,000 SNPs. So on average, each of these 150 different F1 progeny are differed by around 20,000 SNPs, right? So the genotypes of these different backgrounds differs quite a bit. And they also differ quite a bit in fitness, so about 20%. Now, what Milo did was to apply 91 different insertion mutations on each of these backgrounds. So essentially, he constructed 91 times 150 different variants, right? Now you can measure the fitness effect of these mutations, each of these 91 mutations on different backgrounds. And if you plot the fitness effect as a function of the background fitness, you tend to see, as Michael showed, this very nice negative linear trend over here. But of course, this is not the only trend that you see. I mean, there are cases which differ. So for instance, in this case, you see that the fitness effect essentially takes on two different states, depending on what the background is. Or you see cases where the fitness effect is essentially a constant across the different backgrounds. Now, if you, at least naively, what you would expect if you think that there isn't much epistasis, is that something like these two plots over here, right? So you either expect that the fitness effect of a mutation doesn't depend on the background, so you should see a constant fitness effect independent of the background fitness. Or perhaps the mutation has epistasis with one allele in your background. So, I mean, if you plot the fitness effect against the background fitness, you get two different effects depending on whether the allele is present in your background or not. Now, if you imagine that there's even more epistasis, so this mutation that you're looking at interacts with many other loci amongst those 40,000 different SNPs, you would expect that the fitness effect is essentially random, right? So I mean, the naive expectation at least is that this with even more epistasis, this will look like a mess, right? There's all kinds of randomness going on. But what you in fact see is this very clean negative linear trend. So it turns out that across these different 91 insertion mutations, about half of them show this negative trend, right? Some of them do show like a slight positive slope, but there's a heavy bias towards a negative linear trend. Now, the general question that I will initially focus on is to ask what aspects of the fitness landscape actually lead to such patterns. Now, if you think about the fitness landscape and representations of the fitness landscape, one can imagine that, I mean, when we talk about a fitness landscape, we are thinking about a map between the genotype and fitness. So one can imagine that there are many different pathways that contribute to the fitness, and the genotype affects the output from those different pathways, right? So when we apply a mutation, we mutate one locus on the genotype. Perhaps it influences one of these pathways, right? So another mutation can affect perhaps two pathways. And of course, if these two mutations are occurring on the same pathway, and if you apply both of them at the same time, the fitness effect of the joint may not be a sum of the fitness effects of each individual mutation. So there can be epistasis between the different mutations. Now, when we think about many different loci, the picture gets much more complicated, right? So, for instance, in this framework that I'll be talking about, I'm going to imagine that there are L loci on the genome, which defines the genotype. We can imagine that these loci take on two different states, right? I mean, you can imagine that these are two different alleles of the same locus. Now, it turns out that very generally, you can write down the fitness as some combination of the states of the different loci on the genome, right? And this form, this specific form that I'm writing here, has some interpretation in terms of biology, but it also has some nice properties in terms of how you can analyze it. So, so it turns out that these different coefficients over here represent different effects, right? So the first order coefficients, the F's over here, are the additive effects. Specifically, they're the average additive effects when you average over a population where the loci can be plus or minus 1 with equal probability. Right? The second order effects are the epistatic effects. These are the average epistatic effects averaged over the population where the loci take on equal numbers of plus or minus 1. Right? Now,
I have a question.
Yeah.
So here, is it kind of formally possible that you can have like an infinitely fast growth rate? That's, I guess, question number one.
Yeah.
Because I mean, it seems like your negative slope there, if you have a maximum growth rate that you could get, I mean, then you'll have a mostly negative slope from any mutation you get, because if you sort of had a mutation that put you near your maximal growth rate, you're not going to do much better than that. So you'll end up with a negative slope.
Yeah,
but
so there is a possibility that, for instance, that you're essentially what you're seeing is some kind of nonlinearity applied on some additive trait, right? So there's a nonlinearity which saturates at some value, which defines the maximal growth rate. And you can define such models, and that such models have been defined for proteins, and they have been termed I think they have also been termed global epistasis, right? So you have some kind of saturation of the growth rate and you can't go beyond such a value, and that will generate some epistasis. But what's happening here it's slightly different. There's a different kind of effect that's going on here. And I'll show you some predictions that come from the hypothesis that we are proposing. And the structure of the statistics of the effects look a bit different.
Cool. That'd be cool to see. I mean, but surely it is the case that there is a saturating growth rate. You can't divide infinitely quickly. My second question was related to sort of how the cartoon on the top relates to the bottom. So, the bottom, if you have a polynomial and you allow yourself whatever terms you want, you're going to be able to fit a function. And in particular, I was interested in, you mentioned how we can think of some of the terms in the polynomial on the bottom relating to actual biological, things that are of interest. And some of those things are represented as pathways in the cartoon on the top. The pathways, of course, all interact with each other. They're globally constrained as well. So how are you thinking about, sort of the relationship between, in particular, the pathways on top? How are those being incorporated into the model, or maybe it's in some downstream analysis or something?
Yeah, that's a fantastic question. I think that relating to what's happening microscopically at the pathways and how that relates to the actual epistatic effect that we observe, it's quite complicated, right? So because essentially when we look at these epistatic effects, we're looking at perturbations around some parameters that define all the pathways within the cell, right? We can think of these epistatic terms in abstract fitness landscapes that I'll describe later, right? But the question of what these terms actually mean biologically in terms of what's happening at the level of pathways, I think is still a bit unclear.
Cool, thanks. Yeah, I mean, I guess to sum up both my questions, it's that if you write down a polynomial, you'll be able to fit the thing if you give yourself enough terms, but interpreting them as actual interactions, if you've misformulated the problem, for example, because there's saturating effects or because there's weird stuff with pathways, you might be inclined to interpret the higher order terms as
all Taylor expansions are always local though, Brian, right? I mean, so if the question isn't so much whether a theoretical upper bound exists, for example, but whether you're anywhere near it. Would you agree with that?
I guess the point is, yes, so you can always do this and it'll tell you something about the local effect. But
yeah, but expansions like this are mostly informative locally anyway, I think.
That's correct. The caution here is interpreting it as sort of global.
Maybe the speaker disagrees.
Yes, I think there are different representations of the landscape. You can have local expansions, which is like a Taylor expansion around some genotype. This particular representation is a very general Fourier representation of the landscape. There is some specific interpretation. When you think of these coefficients as averaged effects across a specific population structure, right? Which is that these loci are plus or minus 1 with equal probability. But you can have different representations. I mean, this specific one, the reason that we are using it for this particular analysis is that the predictions of this model are going to be tested against the experiments that I described previously. And in those specific experiments, the background genotypes are generated by mating to strains of yeast, right? And when you mate these strains, you get a diploid, you sporulate them, and you end up with a haploid. This haploid is a mosaic of the genotypes of the original parental strains. And you can imagine that there 50% of the offspring is made up of the blue strain and 50% is made up of the red. Right? So for these specific set of backgrounds that we are generating in these experiments, this specific representation of the landscape is particularly helpful.
Okay, yeah, thanks, Gautam. I guess I'd forgotten that we were on the hypercube and that this Fourier Walsh is completely general. You could represent literally any fitless landscape.
Yeah.
That
so
way.
This representation is you're absolutely right. I mean, as you add more and more terms, this is a completely general representation. There is some sense that these terms have to decrease with order. Because if they do not, they're combinatorially larger number of these. So I mean, they should decrease as you go up the expansion, right? But I mean, this is a completely general representation of the landscape. Yeah. So I mean, the reason I like this specific representation is that when we think about the fitness effect of a mutation, all we need to do in this landscape is that we flip the sign of xi, let's say, right? You flip the sign and then you average out over the entire distribution. And there are some nice Fourier properties here. I mean, these terms turn out to be orthogonal, and you can just manipulate things much quicker. But they do have some biological interpretation in terms of averaged epistatic effects averaged over a specific population. Okay, so I mean, I'm going to put out the result in all gory detail, but I'll explain the intuition in the following slide, right? So it turns out that if you take this kind of representation, which is a very general representation, and you make a very specific hypothesis, which is that you assume that there is plenty of epistasis, and that these epistatic terms there are many of them are non-zero, but they're also independent. There are no structural relationships between the different epistasis terms. And we're also going to assume that there are a large number of loci that contribute towards the trait. So it turns out quite generically that the fitness effect of a mutation can be split into 3 different terms. First is a constant, it's just this additive effect of the mutation. The second term is this linear trend that I was showing you before. And the third aspect is a genotype dependent term that it turns out to be a Gaussian random variable. If you're there in the previous session, I mean, Max mentioned something about a central limiting kind of aspect to epistasis when you think about a complex trait. It turns out that something very similar is happening here. And I'll explain a bit more as I go forward. Now, I haven't really explained what the different terms here are. So for instance, this is simply the additive effect of the mutation, of the specific mutation that you're applying. Here it's locus I, right? And the slope of this linear trend seems to have a very specific interpretation. It turns out that this slope, in fact, measures the total amount of epistasis contributed by this locus relative to the contributions from all other loci. So when we measure the linear trend in these assays, what we're essentially measuring is some kind of global measurement of the fitness landscape. It's how much that locus is epistatically coupled to all the loci in the population as a whole. So, I mean, this result, I mean, I'll go to, it turns out that the result is quite simple. There's a central limit kind of argument for this, and I'll just briefly go through it. I think it helps to get some intuition of what's actually happening there. So, I mean, I've rewritten the landscape here. You can ignore the additive terms, they just add a constant. We can imagine that there's a very simple landscape where these epistasis terms are plus or minus ones randomly, right? So many of them are 0, but some fraction of them are plus or minus 1. Right? So if you look at a particular genotype, the fitness for a particular genotype, there are some n plus positive interactions. There are some negative interactions. And so you have some total number of non-zero terms over here. And so the fitness is simply a difference of the number of positive interactions and the number of negative interactions. Now, the key idea here is that the key assumption, the key hypothesis here is that when you mutate this locus, it affects some fraction of all the interactions that affect this trait, right? And also that the number of interactions that this locus affects are many of them. There are many interactions that this locus is involved in. Now, if you imagine what happens on average once you mutate at this locus, what happens, as I was saying, is that you can imagine that you're simply flipping the sign wherever you find an xi. So on average, what you're doing is that you're subtracting out 2 times the number of positive interactions that this locus has, and adding 2 times the number of negative interactions that this locus has. And now if you do some straightforward manipulations, what you see quite easily is that you get this negative trend for the fitness effect, right? So this negative trend is simply coming out due to a sampling bias towards positive interactions. This mutation is randomizing or reversing some of the beneficial effects that have occurred previously. Now, simply by some central limit kind of arguments, the number of mutations that this locus actually affects, I mean, has some binomial sampling effects, and the fluctuations, as long as the number of interactions are large, are Gaussian, right? So what you ultimately get when you apply a mutation in a landscape which has a lot of epistasis, a lot of idiosyncratic independent epistasis, is that you should generically see a negative linear trend, and the other dependence on the genotype should appear as basically a Gaussian residual term. And this kind of intuition comes up quite often in physics, if you're familiar. So for instance, if you look at the expression for the viscous drag on a particle as it's moving through a Brownian fluid, we usually write it in terms of the force is some kind of there's a viscosity that opposes the motion of this particle, but the fluctuations are also dependent on the same viscosity, right? The reason, of course, is that the same molecular effect that generates the viscosity is the one that is generating the fluctuations as well, which is why the magnitude of the fluctuations are related to the viscosity, right? There's a similar effect happening here in the sense that when a mutation is applied, you have many different changes across the genome, across the different interactions within the pathways. And the effect is that you have some kind of resistance. So if there's prior adaptation, you're kind of reversing the adaptation that has happened before. And so you should generally see this negative linear trend. So we can kind of split this into 3 different cases, right? So this is again recapitulating the experimental observations that I described before. The first case is that you have practically no epistasis effects for this locus, and so you should see that the fitness effect is simply an additive effect. It's a constant. It does not depend on the background fitness. If you have one or a few epistasis terms, you should see this kind of 2-state split, where the fitness effect depends on the allelic state of one of the loci in the background. But if you have many different interactions, you have the locus interacting idiosyncratically with many different alleles in the background, then you should see this negative linear trend. Now, in this specific, all of this depends on the specific experiment that we're considering. I think I should highlight that, right? So this really depends on what kind of population we are considering in the background. If we have the experimental resolution and capacity to vary a lot more alleles, a lot more SNPs on the genotype, we should see this effect a lot more, right? Because this effect is going to scale at least combinatorially with the number of loci that you're varying in your population. Right? So, I mean, like as Max was pointing out, this may not be as important when you're thinking of a single biomolecule, but when you're thinking of a complex trait such as fitness, you can imagine that there are actually many different loci that contribute to the fitness. So if you're looking at complex traits, presumably this effect gets stronger as you go up in terms of complexity. I should also mention that a similar argument was proposed recently independently by George Zhang's group in Michigan, where they also observed that simply putting in more of these idiosyncratic epistasis effects gives rise to this negative linear trend. Now, there's a quite straightforward prediction that comes out of this hypothesis, right? So we are saying that this negative linear trend is quite generic. And the prediction is that the residuals around this trend first are Gaussian distributed, which comes from the central limit theorem kind of idea. Second, that the variance of these fluctuations is related to the slope of the fit, which is quite unusual, right? So you should see this negative slope proportional to the residual variance as you do these linear fits. We can test this in the data. It turns out that we can do some kind of independent test, and it turns out that half of the 91 loci that were tested in the experiment satisfy the conditions to have many different epistatic interactions. And for that subset, you do see this linear relationship between the slope and the residual variance. But there are other loci amongst those 91 which do not show that trend, right? So at least for some of these loci, seem to have plenty of epistatic interactions across the different across the genotype. We can also test whether we can predict the distribution of the residuals from the slope. And it turns out that it aligns quite well with the residuals from the data. I mean, so there are some takeaways from this, which I'd like to highlight. The first is that, I mean, the hypothesis here that we're starting off with is that epistasis is widespread, at least for some loci. And the key point here is that it seems to appear in some kind of random fashion, right? So there are no structured relationships between the different epistasis terms, which will give you a completely different effect. But they seem to have a random effect on fitness. Right? And so what's happening here is that a lot of this microscopic randomness seems to be averaging out when you're looking at a complex trait. And so once you average out this complex, when you average out over these different epistasis terms, what remains is this macroscopic trend. And this has been termed global epistasis or macroscopic epistasis. And again, Michael was mentioning some of that from before. The other interesting aspect here is that it essentially gives us some new kinds of experimental assays to measure how epistatically coupled a locus is to the rest of the genome, right? So one can repeat this assay, and it'll give you a sense of how much a locus is globally coupled to the trait. So we can imagine that we can construct different combinatorial experimental assays. So in this specific experiment that I was showing you, there are 2 different strains of yeast that were mated. And so that the background had a specific structure with 40,000 different SNPs, and you have like equal proportions of each of those parental strains. But you can imagine that you repeat the same process with a different strain, and you're going to get a different background. And then when you do this trend, when we measure this linear slope, we get the coupling of this locus not to the same set of SNPs, but a different set of SNPs, right? We can imagine that we can also do some directed mutations on the genome, or repeat the same process in order to measure how a particular locus is coupled to a certain set of loci across the genome, right? So, I mean, the essential point here is that we can use insights from the theory to come up with new combinatorial designs to figure out how different loci on the genome interact with other genes across the genome. So this is at the level of a single mutation. I'll be going forward, I'll be talking about the kind of implications this has for phenotypic evolution as a whole, right? So this is basically what we have from the experiments the state-of-the-art experiments. So what follows is more on the theory side. So if you have any questions here, I think this is a good place to ask about this specific assay and this specific the specific set of predictions that we have here. Okay, so I'll go ahead. Right, so what we had from before is the effect of a single mutation on different backgrounds, right? But what I'd like to do is to go back to how we originally started with, which is to understand whether there are predictable patterns in phenotypic evolution, right? So over the course of a single trajectory, how do these effects manifest themselves when you're looking at phenotypic trajectories over the course of evolution? Now, the classic picture one can imagine when you think about phenotypic evolution is that perhaps there is this effective phenotypic space, right? So you have an optimum within this phenotypic space. The organism starts off somewhere, and as evolution proceeds, this organism climbs up this hill. And some of the observations from Lenski's experiment and from diminishing returns and so on can be rationalized within this framework, right? So as you go up, initially there's rapid adaptation and then you slow down. But this kind of picture kind of gets quite contrived when you think of what happens in the light of the recent experiments which show diminishing returns and increasing costs. And this is nicely depicted in this cartoon that you see here, which appeared in this commentary here. So, I mean, when we think of diminishing returns, what we can imagine is that as you're going up fitness in this phenotypic landscape, the beneficial effects of beneficial mutations is getting smaller. And so you should see this kind of slowing down at the top. But on the other hand, the deleterious effects of deleterious mutations is also getting larger on fitter strains. So there are these kind of steep cliffs as you go up. But as you can see, I mean, this kind of picture is quite contrived. And I would argue that perhaps that these kind of pictures that we're drawing perhaps to guide our intuition are not the right ones to draw, right? Perhaps we can use some of the insights from the theory to kind of re-envision what these phenotypic models should look like. Now, the caveat here, of course, is that I mean, this is biology. I mean, we're going to have a lot of random effects that we can't predict. But the hope is that we can come up with some simple phenotypic models that can guide our intuition, right? So we don't expect these to always hold, but hopefully in some cases we can get some intuition out of these models. Now, when we think about epistasis, the issue gets complicated because essentially you have contingency, which is that you have this very strong dependence on the genetic background. So if we go back to our original simplified picture of adaptation, right? So you have a mutation that arises, it has a fitness effect, which is drawn from this distribution of fitness effects. This fitness effect, this distribution depends on the background. Specifically, it depends on the entire evolutionary history of the organism. So there is this effect that is similar to what you see, let's say, in machine learning, right? There's this curse of history, right? So if you want to predict something which has a lot of history dependence, then the dimensionality of this model is increasing rapidly as this organism evolves, right? So I mean, so we would intuitively expect that predicting evolution gets harder, but I mean, let's go forward and see. I mean, what can we do about this, right? Now, let's say as a hypothesis, we do know what the fitness landscape looks like. And if you want to predict evolution, we can imagine that there are two different approaches, right? So we can imagine that we try to predict the specific, the genotype that will evolve. So we can try to trace out the specific sequence of mutations that will occur along a particular lineage, right? So that is this first approach over here. The second approach is to forget about the sequence and simply look at the phenotypic evolution, right? So we simply track the change in phenotypes over time and keep track of only the phenotypic changes that have occurred in recent history. Of course, I mean, since we know the fitness landscape in this hypothetical scenario, these two approaches are entirely equivalent, right? If there's no other information over here. But it turns out that if you have a statistical description of the landscape, there are advantages to be gained by looking at the phenotypic description. And this has an analogy with what people usually do in fluid dynamics. And I think it's a useful analogy. So if in fluid dynamics, let's say you have a particle moving through a very turbulent environment, right? And you want to understand how this particle changes over time, right? So the analogy here, of course, is you want to understand how the organism evolves over time. So there are two different approaches. They're called the Eulerian and the Lagrangian approach. The Eulerian approach is to specify the velocity field at every point in space, right? And then evolve the trajectory of the particle as you go along. The second approach is to look simply at the statistical properties of the fluid. And you follow the trajectory of the particle as you're moving along the trajectory, right? So you don't actually look at the entire fitness landscape as a whole or the entire fluid field as a whole, but you're actually just interested in the statistical properties of the landscape. So it turns out that when we go into this widespread epistasis limit, the phenotypic approach turns out to be quite a bit more useful and convenient.
Gautam, I think I understand the history now. So if you knew the entire genotypic state, you would only need to know the present. But if you marginalize on something like phenotype, then it's not Markov anymore. Now like the historical states inform you beyond just the present state if you're only looking at like a marginal instead of the entire genotype. Do you agree with that?
Yeah, absolutely. Right, so this is it, right? So I mean, like we're going to try and build a frame. I mean, the entire, I guess, the entire point of this analysis is essentially that we have these experimental observations that Michael showed and that we saw, and we are trying to build this framework where we can simply work at the level of phenotypes, right? We can abstract out what's happening at the sequence level and simply look at what the changes that are happening at the phenotypic level and hopefully give a statistical description of what's happening at that level. And that's essentially the broad picture. So we can imagine, I mean, we came up with this framework. Maybe, I mean, perhaps it's not the best way, but this is something that we found that was useful. One way we can imagine is that in the lab, we are measuring some randomly picked genotype, right? We measure the, we go through an evolution experiment. So this strain shows some sequence of fitness changes. And now we'd like to ask, what's the distribution of fitness effects for the different loci, conditional on the sequence of fitness changes that it has gone through, right? Now, I'm going to argue that when you have widespread epistasis, you have a complex trait, and you have epistasis that is essentially idiosyncratic, and you do have a lot of, you have many loci that contribute to the trait. This distribution of fitness effects is essentially a Gaussian distribution because of these central limit theorem kind of arguments. And so essentially you can write any fitness effect as a linear sum of all the phenotypic changes that have happened in the past, right? So you can predict something related to, based on what you have observed. And of course you have some uncertainty because you don't know exactly the genotype that you have picked, even though in this specific framework, we do know the landscape. So the key thing over here is that when you expand in this fashion, the coefficients that quantify this history dependence are not the microscopic ones, but in fact the macroscopic ones. These are quantities that define global features of the fitness landscape. These are not the microscopic fitness features. To give like a few examples, right? Like let's imagine that we truncate this expansion within the first term. So you're simply looking at the fitness effect conditional on the current fitness, right? So this one-step history dependence turns out to be exactly the linear trend that I was talking about before, where you see this, the fitness effect has a linear dependence in the background, and the slope is related to the total epistasis contributed by this locus related to the full trait. When you look at two steps, right? So you have one mutation, and then you're looking at the fitness effect of the subsequent one. It turns out that you again see this linear relationship between the current fitness, but then you can expand this further and you have a dependence on the fitness effect of the prior mutation. And there are some complicated dependencies that come about here. But the key point here is that these coefficients that you see here are global features of the landscape. They're related to the microscopic features, but they end up being some complicated combination, some complicated sum of all the features that contribute towards the landscape. And of course, these are the averages. I mean, you do have plenty of fluctuation. So you can't predict everything. There is some inherent randomness over here. Now, these coefficients will depend on the specific landscape. And the question that we would like to ask subsequently is, are there certain specific fitness landscape architectures that give you quite a kind of minimize the dependence that you have on your history, right? I mean, this is kind of what we are seeing in the Lenski experiments and from the evolution experiments that people have observed. So it turns out, I mean, you're seeing that most of the fitness gains seem to be dependent only on the initial fitness. So the question is whether we can come up with some architectures that do have this property, right? This property that most of the fitness changes depend on the current fitness, and not on the entire evolutionary history. So it turns out, luckily, that there are certain architectures that do have this property. And I'd like to say that, imagine that we cleverly constructed this one and we found this, but in fact, it was by total chance. It just turned out to be something that we tried and it worked out. So in this model, what you're seeing here is a specific fitness landscape. This landscape, you're imagining that there are many pathways that contribute towards fitness, and each locus affects some fraction of all the pathways that contribute towards fitness. specifically, we're assuming that you have this kind of representation of the landscape that I was showing you before. We are assuming that the epistasis terms are proportional to the probability that the different loci that contribute to that epistasis terms interact, right? So for instance, if this locus contributes towards some fraction of all the pathways, the probability that some set of loci will interact is the probability that they all are part of the same pathway, right? This captures that intuition that you have many pathways, you have loci that contribute towards many different pathways at the same time. And so you will have epistatic interactions across different loci. So it turns out that this specific model, there is some intuition. We have some intuition for this, but the derivation is quite technical. It turns out that for this specific model, the fitness effect of a mutation depends only on the current fitness. So there's some kind of Markovian property for this specific fitness model. So you can imagine that if you want to study the evolution on this fitness landscape, what one can do is to pick a mutation. You draw this VI, which turns out to be related to the sparsity over here, from some distribution that quantifies the architecture of this landscape. We draw epsilon, which is a Gaussian random variable, and so we get the fitness effect. Let's assume we have some selection dynamics, and so we can study how evolution will happen on these kinds of landscapes, right? It's like a stochastic dynamical system. And notice that most of the information about the microscopic features of the landscape is essentially gone. There are many pathways that lead to higher fitness. It's just that going up fitness gets harder as you gain fitness. So we can generate different trajectories coming from this model. So here I'm showing you 10 different simulations generated from this model in the mean in this yellow solid line. And I'm overlaying this over the data from the Lenski experiment. There are 3 parameters here. There are simply 3 parameters, and these 3 parameters control where this y-axis is located, and you can also scale these different axes in different ways. And here I'm saying it's qualitatively consistent because the specific assumptions we have made are quite different from what's happening in the Lenski experiment, right? The selection dynamics, as Michael was saying, are quite complicated. We have assumed some very simple selection dynamics, but at least on a qualitative level, it does seem to be consistent with what we're seeing in the data. So we do capture this rapid increase in the beginning, followed by the slow and steady rate of adaptation as you go, right? And I mean, when I first saw this, I was surprised because I mean, it's one curve with not too many free parameters. So I mean, to some extent, we are perhaps capturing some of the right effects. I mean, I guess I'm running out of time, but I'll just say that the key conceptual idea that perhaps that we are proposing here is that when there's a lot of epistasis, one, instead of thinking about a phenotypic model where you have a single optimum and you're going up this optimum, perhaps a better way to think about this is that you have a notion of adaptedness, right? So you have basically organisms that, as they're evolving, initially they are neutrally adapted to some environment, but as you evolve them, they get more and better and better adapted to the environment, and this restricts the number of paths that they can take up the fitness landscape, right? But this adaptedness is a key quantity that determines how much they can evolve into the future. And the intuition of why this specific shape comes about is that initially you're neutrally adapted, so this linear negative effect is not there. Mutations are not reversing the effects of the previous mutations that have occurred over the course of evolution. So they're essentially having random effects. But as evolution proceeds, it gets harder and harder to find beneficial mutations. So the distribution of fitness effects, as Michael was showing, does have some shape, but as evolution proceeds, this distribution shifts towards the negative side, right? but it in fact shifts in very stereotypical ways, at least in this specific model that we're considering. So, I mean, it just so turns out, I mean, I'm just going to put this slide here. It turns out that in this specific model that we're considering, there are certain universal dynamics that you should see, expect to see. I mean, for instance, it always turns out the fitness rapidly increases linearly in the beginning, and then you should see some kind of power law scaling as you keep evolving this organism. And another aspect is that if you look at the distribution of fitness effects, so not just the beneficial ones, but also the deleterious ones. Initially, when you're neutrally adapted, you should see a symmetric one. But as you become more and more well adapted, which is this y greater than 1, you should see a shift. You should see this kind of shift towards beneficial mutations, right? But as Michael was saying, I mean, the specific fitness effects of the different loci are all moving around within this distribution. It's just that the distribution remains roughly the same given a particular amount of adaptiveness. I mean, the general idea here going forward is to imagine that, I mean, we looked at one specific landscape and we got one dynamical system essentially, right? So, I mean, in reality, I mean, we are not going to have a fully connected landscape for the cell. Presumably, we have different modules, probably different modules that contribute, metabolize different sugars or so on, and all of these together contribute towards fitness in a particular environment. So the question is whether different architectures lead to completely different phenotypic dynamics, right? And we have this framework which we can use as a hammer to analyze what kinds of evolutionary patterns will come out from different architectures. The second question is whether we can actually infer these architectural features from genotype-phenotype data that you can measure in the lab. I think because of, I mean, the framework gives some specific quantities that are important if you want to predict phenotypic evolution. These are global features of the landscape. And so we have a sense of what to look for when we actually measure these genotype-phenotype maps. And the third aspect is that I'd like to highlight is that the importance of combinatorial designs, right? Combinatorial designs essentially give you a way to probe global features of the landscape rather than just looking at locally around a single genotype. And I think these give you complementary information. I mean, of course, the local ones are also important, but these give you a different kind of information, and this information might be helpful if you want to look at things at the phenotypic level and not just at the level of individual sequences. All right, thank you.